Hello everyone. Today we are going to start with a new subject in Elma. Previously we had done maths and computer applications of both ICSE and CBSE. We are going together. Mm, Today we are going to come up with biology. Today's class would be on biology class 10, chromosomes and cell cycle. We will be doing it in some parts. The full chapter cannot be done at once because it is a big chapter. Now we will be beginning with today's session. It will be the first session for biology in ILMA and it is of class 10. So let's begin. Chromosomes are present in the nucleus of a cell. Now in an organism there are many types of organ systems. Organ systems compose of many types of organs. Organs contain many types of tissues. Tissues contain many types of cells. And cells have cell organelles within them. Within the cell there is a nucleus which will have some chromatin. If the cell is not dividing in a normal cell, the beginning cell or the cell which is just born will have only chromatin and some nuclear matters like nucleoplasma and nucleosome. Now that chromatin during cell cycle at the phase of division will thicken itself and divide into two halves. That would be the chromatin will come together, it will condense together and form thick fibers which will be called chromosomes. Now within the nucleus of a cell, nuclear membrane in animals are not continuous, they are discontinuous. There are chromatins like very unmanaged, discontinuous, single long thread-like. They are very fine thread-like structures present inside the nucleus. They contain genetic matter such as the DNA. We will be studying that in today's class. Now this chromatin will condense up to form chromosomes, which will be basically in the form of X-shaped structures as we had studied, which will be depicted as like this in the diagrams. This is a chromosome. Now a duplicated chromosome. This chromatin fiber will form a chromatid from this half of the chromosome. This and this together is one single chromatid that will be duplicated during the cell division, and it will form another half of itself which will be together known as a chromosome, duplicated chromosome. Now this chromatin has DNA within it, DNA and other genetic material. Now the DNA means deoxyribonucleic acid. Now it is the first time we have been exposed to the specific elements of DNA. For basic class 10 we will just get a basic information of DNA. DNA will have nucleotides within it. Now nucleotides are composed of three elements: phosphate, sugar in the form of pentose sugar, and another substance is nitrogenous bases. Now it will be formed in the structure like in DNA. There are phosphate. This is phosphate. It will be combined with pentagonal shape of. It will be combined with pentagonal shape of sugars, which is sugar pentose. Now this sugar will be combined with nitrogenous bases, and the same thing will be on the other half. Some nitrogenous bases will have a shape like this, others will have a shape like this. To combine together the nitrogenous bases, this is an important question, but sometimes you don't keep focus on it. These nitrogenous bases of two different nucleotides this complete complex is a nucleotide, where this is phosphate and this is the nitrogenous base, and these are also of many types. In DNA there are 3 types. Now this complete complex is a nucleotide. The same complex will be on the other side. The nitrogenous base will only be a little different. These nitrogenous bases will be combined with a hydrogen bond. This is an important question. This diagram won't be coming in the examination, but it needs to be studied. Now, nucleotide and nucleosomes are two different words and have very different meanings. I will be coming to nucleosome. When I come to that, I will be showing how to differentiate between nucleotide and nucleosome. Now, the nitrogenous bases. Coming down to the nitrogenous bases. They are of 3 types in DNA. One is adenine, guanine. Oh, 4 types. Guanine, cytosine, thymine. Now, this type of question might be asked in diagram-based questions. Adenine will combine with thymine, guanine will combine with cytosine. They are represented as A, G, C, and T in diagrams. Now, if A is like this, then thymine has to be like this to form a complete shape. This bond present in between is the hydrogen bond, and if guanine is like this, if guanine is like this, then cytosine will be like this to form a complete shape between which is the hydrogen bond. Now, these are the 4 nitrogenous bases. They are found in repeating sequences in a DNA molecule. DNA is a double helix structure having 2 strands in it, meaning that this is a DNA molecules A and T, G and C, E and T, G and C, and so on. Now, adenine and thymine. Yes, adenine and thymine will be combined together in 2 bonds, and guanine and cytosine will be combined together in 3 bonds. 2 bonds and 3 bonds, like in chemistry we had done organic bonding. Now there are 2 bonds in this and 3 bonds in guanine and cytosine. There will be repeating sequences in a DNA molecule, double helix. This is a double helix DNA molecules, and it is known as a ladder-like sequence because of this ATGC, AT and GC. These are the steps of the ladder, and this is the ladder itself. A complex of this is the DNA. Now, the picture diagram which I had shown was a diagram of a nucleotide. Now coming to the histone proteins, the chromatin, I told that the chromatin was compacted, condensed into chromosomes. Now how they are condensed are because of some proteins which are known as histone proteins. Histone proteins are 8 in numbers. Therefore they are also known as histone octamer. Oct meaning 8 and histone are the name of the proteins. Histone octamer. Now coming to that. Suppose this is a chromatin. The histone proteins, 8 in number, will condense this and form it into a ball-like substance. Now, this is known as a histone octamer complex, which is also known as a nucleosome. This is also referred as a football-like structure. It will be like the footballs. The histone protein form a ball-like structure, and because they are separate ingredients, like a football, we get this hexagonal shapes combining together like this. Now these are the this shows similarity between the histone combining and the football complex combining. Therefore, they are also known as football-like structures. Now, these histones are these histones together with the chromatin will condense them, and this complex is known as a nucleosome. And I was telling how to differentiate between nucleosome and nucleotide. One might forget the difference between them by the names. Now that was yes, I was nucleosome and histones. Sorry, no, nucleosomes and nucleotides. Nucleotides. The name nucleotides. This is the spelling. Has three components within it, and this T, which will refer as 3, will show us that this T we need to find out. If the name has T in it, it will be a 3-component structure, which is a nucleotide. And if the name does not has T and it is named as nucleosome, then it is not that 3-component structure, it is this 8 histone protein structure. Now this is how we differentiate between nucleotide and nucleosome, just by the help of this T. In many other words in biology and other subjects, we can use this type of format to differentiate between two similar spelling words. And now going to the next part, which will be structure of chromosome. This will be the final thing we discuss about in today's class. Now a chromosome is somewhat like this. This is the centromere. And these are the chromatids. These are sister chromatids because they are duplicated ones. I told earlier they are duplicated, therefore they are known as sister chromatids. These are chromatids of the same one, therefore not sister. Chromatids. Now this centromere joins the two duplicated chromatids together and form a single chromosome. This chromosome, as we had studied in the lower classes, they are basically this X-shaped structures, except the Y chromosome and some of other few chromosomes. This like the mutated ones have a different structure. The Y chromosomes is the smallest chromosomes of all. Now this X-shaped chromosomes will have a centromere joining the two chromatids together. Chromatids which are condensed form of chromatin, and will have nucleotides which is within the DNA, which is within the chromatin, and will also have nucleosome because they are condensed with the help of histones. This is the complete thing we had studied. Our aim was to study in today's class. So this would also have some genetic materials within them. Therefore, they are also the carriers of genes. And the cells, these are the present in the nucleus during division state, not during the normal state. In the normal state, there are only chromatin. In the division state, there are chromosomes. Now this will contain the genetic material. Therefore, we say that the nucleus has all the genetic information. This in the nucleus has all the genetic information. Now this would be the end of today's class. We will be doing the further topics in this chapter, like cell cycle, cell division, the different phases, anaphase, telophase, prophase, metaphase, and all cytokinesis and chirokinesis. So this will be all for today's class. Please do like the video, share, subscribe and follow, and do post your queries on any subject in the three sciences and maths and computer application in the comments below, and I would be looking forward to solve them out. You may WhatsApp me at 98749445154 to send your queries or any other information. So thank you.